I'm Tina Han. I'm the Senior NGS Business Solution Manager. I will present sequence with confidence, how long-read capture with Twist target enrichment system can empower your research. So Twist have a special way of making oligos. So we don't synthesize in a 96-well plate. We are using a silicon wafer to make 9,600 genes in one go. So this really scale up the synthesis process. And when we synthesize the oligo, we have very nice GC profile for the synthesis, and then we will use a proprietary way to amplify it up, so we have enough to do the capture. And because we have this special way to amplify, then we can maintain this GC profile in the your capture performance. So then you will always get the very nice uniform capture in your data. In contrast, our competitor tend to have bias during their amplification method, so they will have a lot of valley and and peaks that are either under sequence or over sequence. So our way of amplification really makes sure that you have a uniform data at the end. The other advantage with Twist is that we have a double-stranded DNA to capture your region of interest. So our double-stranded DNA can catch sense and antisense of your region of interest. So you can think of it as like 2 shots on goal. Instead, our competitor only are having single-stranded DNA that can only capture your DNA of interest once. The other unique way of Twist target enrichment is that we have all the blockers and probes and beads are in our solution. So we have, once you prepare a library, we are using blocker to block the adapter sequence and also the repetitive regions. And our probes are biotinylated, so you can use strapping beads to capture it using a magnetic bead system. And then during some washes, then this library fragment are ready for sequence. Normally, we look at the target enrichment performance metrics by 3 items. So uniformity, specificity, and library complexity. So again, uniformity is something that if we amplify the oligos during synthesis is already uniform, then it will also translate into your data. So you don't have a lot of over sequence or under sequence region. That translates into sequencing economy. So the more uniform your data is, then the less sequencing you have to do. Specificity-wise is that how much probe we have in your capture can target your region specifically. And library complexity is something that you want to maintain the highest as possible, just because normally the bioinformatics pipeline, the first thing they kick out of your data is the duplicates. So you want to make sure that you can maintain the highest library complexity as possible. So this slide kind of summarizes that in order to present or to preserve the useful data in the green, you want to minimize the over sequencing area, also want to minimize the under sequencing area and also the off-target region. So you can get the most information with the least amount of sequencing. In summary, Twist target enrichment can bring you exceptional performance because we NGSQC all the probes. So you will have no probe dropout when you get the probe in the shipment. And we have very flexible modules, so you can order different kits, reagents separately. And last but not least, the whole workflow enables you maximize sequencing efficiency. So you will use least amount of sequencing to get the data you need to do variant calling. So just quickly go over 2 fixed panels. So we first have the SARS-CoV-2 research panel that at the start of the pandemic in March 2020, we have launched this panel. We don't have to ever change the content just because our probe are so special design that they can capture even with mismatches. So this means we can still capture the new variants. So for example, the Delta variant, Omicron variant, you name it, we can still use the same panel to capture the latest variants. And the same with our comprehensive viral research panel, that is a larger one, so you can capture all the variants you want to see. So we have shown that you can capture Zika, you can capture monkeypox. So again, our way of probe synthesis and plus our design algorithm really maximize how much you can see in your data. To summarize, the key benefits of target enrichment is that instead of using whole genome sequencing, that you will use 90 gig of data to see just a few reads on your regional interest. But now, for example, in this case, exon capture, you capture all like exons in your data, but you only need 7.5 gigabases. And then you can see the regions very well. So you will normally get more than 20 reads in the locus, in contrast to 9 reads in a whole genome sequencing example. So next, I'm going to go into detail for the long read capture application, just because with PacBio, we have collaborated on several projects, and I'm going to go into deeper details. So here is an application of showing how Twist target enrichment coupled with HiFi sequencing can bring best out of the data. For example, here are just 3 panels. Example, you can have a large panel on a very large scale, 20 megabases, about 400 genes, and you can put 4 samples in one SMRT Cell. The medium panel about 2 MB, then you can put around 50 genes in one panel, and then in this case, you can have 24 samples in one SMRT Cell. And for the small panel size, 100 KB, for example, BRCA1 or 2, you have only 2 genes because they are very long genes. And then this example, you can have about 96 samples per SMRT Cell. So together with the Twist and HiFi sequencing is that you can put more samples in one SMRT Cell. That's mean, you lowers the cost of sequencing dramatically. And we have shown that with Twist target enrichment, you can get a very good resolution of your target of interest. This example is showing the NCF gene that's associated with chronic granulomatous disease. That in the HiFi WGS, you have quite shallow coverage. However, with HiFi target enrichment, you can easily see the high enrichment with different haplotype, and you can fully phase them. In contrast with the short read, then you will tend to have gaps, right? Because due to mapping issues with the short read technology. And we have another example showing, in this case, is a DPYD haplotype, that if you capture the regions, this is a very long genes as well, and with a lot of exons, that you can, again, fully phase the different haplotypes, and we can even do star allele calling in concordance with the G-term consensus call. And over 50 variant position were phased nicely in this 50 KB region of DPYD. And we do have a long read capture protocol that's available online on our website. It is compatible with PacBio SQL2 and SQL2e. And this slide just showing you what are reagents, and the panel are compatible with this Twist-PacBio workflow. So all the boxes highlight yellow are recommended for this workflow. And just to start from the beginning, that the panel design, when you come to us, if you submit the list of coordinates or gene names, we will turn around the design within 1 to 2 days. And you will take Twist to synthesize the panel within 3 weeks. And then you can carry out the genomic DNA extraction fragmentation that takes about 1 day. and you do the library prep for 1 day, and target enrichment, you will normally start doing overnight, and next day would be just only 4 hours to finish up. And then you can actually have the afternoon to do the PacBio library prep, and then start the sequencing on the same date. And then within 24 hours movie time, the next day you have the data for analysis. And the third-party reagent we recommend are the KOD polymerase and DNA strengthening beads that are with larger diameter. And here's just a case study with our customer from Leiden that they have used our Twist hybrid capture panel, that they have about 23 genes in this 2 MB panel. They have a lot of CYP genes, HLA genes, class 1, class 2, and also other pharmacogenomics pharmacogenes also in this panel. And again, this is the whole workflow showing you how the wet lab from the sheer DNA to library prep to enrichment, and then to SmartBio library prep, and also the pipeline via the SmartLink. And then for variant calling, that's PacBio already set up in their pipeline as well. And here is a nice result showing that when you have 24 samples in one smart cells, you can get very high coverage, and about 94 of target region are at least covered 30 times. So that means you can do a variant calling very confidently. And the long read capture application, so far we have been working with various customers. We have working with pharmacogenomics panel, HL panel. We also have done a lot of inherited disease panel that focus on neurodevelopmental disorder, for example, FF1, or whatever genes is difficult to capture with short read. We all demonstrated it can be easily done with long read. And last but not least, we also have done some proof of principle analysis on repeat expansion and also methylation, just because sometimes the customer may not want to sequence the whole genome because that could be very costly. If they only have a certain promoter they want to enrich, they can easily couple with our target enrichment system that they can sequence those regions very deep, like above 200x. So then they can make sure what they are want to see is there. We also see a lot of customers trying to consolidate a lot of empty-con assay. For example, they have done with single gene, and now they can merge all the empty-con into one assay. So using a capture approach, then they can do more than 50 genes in one go. So that have been also very efficiently try to scale up, try to make their lab more efficiently. So this is something that you can also think about. And last but not least, either you could want to look at viral genome or viral integration. For example, you want to see where integration site is. Maybe you need to have a long piece of DNA in order to find it. Then you can also use the long read capture. And we see a lot of AgBio are also using the long read to try to capture the genes that were previous not, the genome wasn't unknown. So they would need the novel assembly, but then they could actually try to start with cDNA capture so they know all the exonic regions, and then use that to try to capture the region of interest on the gDNA level. And with cDNA, we have seen a lot of customers doing isoform detection and fusion detection because they can also try to capture the known or novel fusions. And also the rare transcript detection, just because now you may, if you want to do the whole transcriptome scale, you may lose the resolution on the lowly expressed genes. So you want to see those lowly transcripts, you can also do it enrichment, and then you can fish them out and then try to interrogate deeply. And since our algorithm and probe design and our capture technology, we are hypothesis neutral. So this way can allow you to see a lot of regions that you previously are not able to see, or not probably the fusion were unknown in the literature. We welcome you to talk to us. So you can either send an email to our customer support at twistbioscience.com, or you can catch us at the ASHG. It's coming out at the end of October in Los Angeles, or the long read sequencing in Uppsala, just right after ASHG. So now I'm welcome to any question you have. Thank you for your attention.